You get stuck, Adriana. What's
Wait,
happening?
now it's
Okay, now it's working. Adriana, you're muted. Adriana, what's happening?
Now it's
Okay,
Now
it's working.
Now everything is working. Sorry. Okay, so let's get started. Good morning, everyone.
Adriana, you're muted.
Again, but I can't.
now it's
Now everything is working. Sorry. Okay, so I need to mute the YouTube because I'm hearing myself all the time. Okay, so good morning, everyone, and good afternoon, or good night, whatever you are. It's a pleasure to have you all here. And before we begin this seminar today, I'm gonna thank for this awesome talk given by Dan Rubenstein last week, which we heard about horses, zebras, and wild asses, and the causes and consequences of behavioral variation and flexibility. Before we start our seminar, I also want to remind next seminar speaker is going to be Lucy Aplin. She's going to talk about culture and cultural evolution in birds. So don't miss out, stay tuned. And today we have Dr. Hanja Brandl. She's going to be talking about stress and stress transmission in social groups. Dr. Hanja Brandl, she is currently a postdoc at the Max Planck Institute of Animal Behavior, where she's been since 2018, if I remember correctly. She got her PhD at the University of Hamburg, and then after that, she became a postdoc at the Max Planck, where she's been very successful acquiring funding for her research in social transmission of stress. She's currently working with zebra finches, but also she's extended her collaborations to work with wild systems, looking as well into early determinants of social stress. And also, Hanja has been very successful in her publication record. She's been publishing in Journal of Animal Ecology, Endocrinology, Methods of Ecology and Evolution. She's very interested in looking or understanding the early life determinants of social dynamics and collectives. She takes into account different approaches, physiological and I'm sorry, I have to look into my notes because she uses a lot of approaches: physiology, evolution, behavior, and ecology. And she also combines not only lab experiments, but also field studies and large-scale automated data collection to look into the fine scale physiological environment of the systems that she works with. So with this, and no further ado, I would like to give the mic to Hanja. Hanja, please. You can share now.
Thank you very much, Adriana, for this nice introduction. Let me try to share my screen. Can you see it well? Can you hear me well? Everything working? Okay, great. I'm gonna try to make the video small so I don't everybody in the way of my slides. Okay, great. Yeah. Good morning, good afternoon. Yeah, thanks for the introduction again, Adriana, and thanks for the invitation. It's really been a great pleasure for me to be here today. I am feeling a little bit like a small fish in this seminar series between a lot of quite prominent established researchers who've done a few more years in this business already than me. But yeah, as Adriana was already struggling to list all the different methods and approaches I've been working with, it's true that I've also already done a few things. So I'm very excited about the opportunity to tell you a little bit about my humble work today. Yeah, so something that I have become very interested in my research is physiological interactions and how they shape social dynamics in groups. So I think that physiological processes are really powerful and impactful in shaping many other behavioral processes, including social ones. which probably many here will agree with anyway. But I also really think that there are a lot of interactions and dynamic happening in social groups involving these physiological processes that have not yet been so well explored. So if you think, for example, of the stress response that sets in when our system perceives danger with the classic fight or flight response, probably the most prominent example of a strong physiological response. We've all experienced at some point in our lives the adrenaline rush that we get in the situation that our body perceives as an acute threat that tries to mobilize energy in our system. So we've all somehow been this gazelle in one way or another, even though we very rarely get chased around by a cheetah these days. But it might just be a car with a reckless driver cutting you off in the morning traffic, or fast approaching deadline that makes you wake up at 4 a.m. and shiver, that also triggers the stress response system. So the stress response is obviously involved to increase our chances of survival, but there's also a balance between the cost and benefits of the stress response. So if it achieves that, if it actually helps us to survive, great, that's really a big win. But the stress response can then also incur costs for the organism, as the stress response is also physiologically demanding and can lead to wear and tear of the system. And especially when stress responses are triggered constantly, so when the stress becomes chronic, when stress responses are triggered repeatedly, which can also lower the threshold for that to happen next time actually, or when there's a mismatch between the stimulus and the response. So this has traditionally only been measured at the individual level. However, in social animals, we might also want to start taking group level outcomes into account. So because stress might really affect the performance of a social group as a whole, and it is relevant because many or most animals spend at least part of their lives associating with others. So seabirds, like the gannets, for example, they spend the time out of the sea mostly alone, but then they come together for reproduction in these huge breeding colonies. We can think of hyenas or dolphins as examples of fission fusion societies that have very long-lasting social bonds, but they can also spend time alone or in pairs and then come together to hunt or eat or play. Meerkats are an example of highly social, cohesive groups that stay close together most of the time. Or we can think of fish swarms and bird flocks where social interactions lead to this highly coordinated collective movements and actions. So my point here is really just that life is very social and collective for many animals out there. And the role of stress in the context of sociality is still fairly unexplored, I think. So just to give you a quick outline of what I'm going to talk about today, I'm actually just checking the comments. Can you hear me all right? Is the sound quality good? Okay. Just have a look in between that you're actually hearing me when I'm talking. Can you see my mouse pointer as well? Okay, cool. So my outline today, so first I'll start by giving an introduction to stress and stress transmission. Then I will talk a bit about the social system of zebra finches, which was one of my main study systems so far that I've worked on. I want to introduce a little bit the empirical work on stress transmission in birds, and then just give a little bit of a wrap-up outlook and maybe bring up some open questions to discuss. Good. So just a very quick recap about the stress response. If you want it or need it, I won't be going too deep into these details for the rest of the talk, but just a quick summary. So as I already mentioned, the stress response helps organisms maintain physiological homeostasis, broadly speaking, when they're disturbed by an environmental stimulus that we call a stressor. So a stressor can really be a wide range of things beyond the classic predator example. It can be social stress like aggression, giving a public talk, be a form of pollution, starvation, rising gas prices, so on. There are a lot of options what can be perceived as a threat in this ever-changing and rapidly evolving world. So the stress response really starts in the brain that discriminates what even is a stressor to us, because not everything is equally stressful to everyone, and this might also change between different time points. So this involves the limbic system, the amygdala, and then the hypothalamus. It's orchestrating a lot of the downstream processes involving hormones and metabolic processes. So the stress response does involve multiple parts and multiple systems. It is on one hand the autonomic nervous system, triggering a fight or flight response. So the autonomic response acts really fast and involves, among other processes, release of adrenaline that then leads to heart rate increase, and so on. And another important stress system is this endocrine cascade via the HPA axis, the hypothalamic pituitary adrenal axis. This is a much slower acting response which triggers the release of cortisol and corticosterone as the major glucocorticoids. So these are metabolic hormones, very important in everyday metabolic functions. But they can also help mobilize energy during a stress encounter by increasing blood glucose. However, they only really peak in the bloodstream after 20 or 30 minutes. So then the role, if the stress has passed, can also be involving in bringing down the stress response via negative feedback. But if the stressor continues to be present, the glucocorticoids can also try to help balance the energy demands of this continued emergency situation and prepare for longer response. And ultimately, we can also expect to see various behavioral adaptations in response to a stressor. So where individuals may exhibit some form of stressed behavior. And the stress system like this is actually very similar across vertebrates, at least with some differences. Like, for example, the dominant glucocorticoids, corticosterone in birds, amphibians, fish, and some rodents, and cortisol in humans and many other animals, mammals. Yeah. So this is all complicated enough and still a very simplified field of things, but we can see the different parts and aspects of the stress system are involved with different time scales. And throughout the rest of the talk, I'm sometimes talking a bit generalized about the stress response, or just stress or being stressed, which is sometimes because I don't even know what systems exactly are involved in a certain process, but mostly it will most often probably be referring to mid or longer term activation of the stress system, because this is also much more accessible to study. So the adrenaline response is so fast, for example, that it would be even really hard to measure it. Yeah, so that said, when we measure any interesting physiological parameters in individuals, but we are looking at social animals, as most animals are, I think it's also interesting and important to consider that the physiology of the individuals in the group can also interact with each other. So I will give you some examples of where physiological interactions between individuals take place. There are many examples from humans, for example, where individuals match their autonomic functions like heartbeat, breathing rate, or skin conductance with a person physically close to them. So this can occur between complete strangers, but even more so between close social partners. And the level of synchrony was even found, for example, to be an indicator of attraction between individuals. So during a first date, the synchrony in heart rate and skin conductance between two people are actually better predictors for the attraction between them than more word signals like smiles, laughter, or eye gaze. You might want to start checking the skin conductance of the date. Yeah, I think it's interesting because these physiological processes are obviously happening unconsciously and are very difficult to regulate. Also in larger groups, that physiological synchrony was actually linked to the levels of cohesion and performance of social groups. So interpersonal synchrony is pretty widespread on different levels across both humans and other animals. But also on these physiological levels, I think that physiological synchrony might facilitate social bonding, cohesion, and interaction in groups. To move to a more animal example, we can look at breeding colonies of birds, where large numbers of individuals often show a remarkable level of synchrony in the breeding activity. And so on one hand, this might obviously be mediated by environmental factors and are perceived by everyone, like photo period or food availability, but it was also suggested that synchronized breeding may be coordinated by responding to physiological changes of the breeding neighbors. So that could be simple cues like agitation or stress levels going down in the neighboring pair, or reproductive hormone levels going up in the neighbors, that can then serve as a social cue to adjust their own hormonal levels. And this response may then propagate through the colony, so that just these small local adaptations then help to achieve colony-wide synchrony. And another example is stress transmission, so where the stress response of one individual becomes activated after interacting with another stressed individual. So the observer triggers a stress response that can be as strong or almost as strong as that of the individual that was affected by the stressor directly. But the observer has never even experienced the stressor firsthand. So stress transmission is something that I've become particularly interested in, and it's something that has also been studied and acknowledged in humans since the 70s already. So most often in pair relationship or marriages, so where the stress of one partner, for example, at work affects the other at home, which I think is quite comprehensible and relatable, but it has rarely been investigated what dynamics this could have in larger groups of people. So in the work that some of my collaborators are doing on humans, so they induce stress by a staged interview situation, which is really pretty mean because we get this really unfavorable interview panel that is not giving you any positive feedback. And you're just standing there trying to present yourself, and then on top of that, you have to start counting backwards in steps of 23. So this is actually quite reliably inducing stress in most humans. So this is known as the triosocial stress test. And then we can measure the stress response of the test subject by measuring hormones in the saliva. So in the people who are direct subjects to the test. But then they also have other people observing the subject getting more and more stressed. And some of these observers then responded with a weakened stress response or even a stress response nearly as high as that of the test subject itself. And so the next step here might be, for example, to test how the group of observers then performing group tasks after being exposed to this indirect secondhand stress. There's also quite some evidence for stress transmission happening in rodents. So some of my other collaborators are investigating this further in mice. But no one has actually looked very much at this in other vertebrate groups, like birds, which is what I'm doing now. And I think that birds make a great study system here because they do show a lot of interesting social and collective behaviors where we can study how this gets modified. Group members are stressed. But not only in birds, but in general, it is still widely unexplored what the consequences of such physiological interactions could be for animal collectives. Until the stress response is actually evolutionarily very deeply rooted and very similar across vertebrates, it's probably very likely that stress transmission could be widespread across these different social vertebrates. So to explore this, we have this interdisciplinary project on stress transmission at the Collective Behavior Cluster in Constance, where we're working on different animal groups to explore this further. I want to talk about talk through two examples of other studies that have done before I started working on the subject, just to give you a bit of a background on the existing state of the field. So the only study actually that I know of really that has been done on some form of stress transmission in birds. So that was done on these black-legged kittiwakes, actually on chicks in the nest. So they had nests of 3 chicks, and one of them, each and every in half of the nest, also one of them received a corticosterone implant, so a form of stress manipulation. And then they basically measured what happened to the other chicks compared to control nests without a stress manipulated chick. And so they showed that the plasma corticosterone levels of the so as in the dark orange bar, the stress implanted chick was almost as high in this light orange bar, which was basically the siblings of the stress implanted chick compared to control nests without a stress manipulated chick. And they also measured some other behaviors, for example, like here we have the time to crouch, which you see on the picture on the right, the time to crouch in a predator response. So the stress-treated birds were faster to respond to a predator. And they also showed that the stress-treated chick and the siblings all grew slower, had weaker plumage, and yeah, there were several measurements that were affected by this. Yeah, in this case, I don't know, there was a bit discussion about this work. Also, it could be, for example, that the parents mediated this effect, that were basically the parents were affected by the stress manipulation. So we don't know exactly what's happening, but I think it's very interesting, and it's also something that really got me interested into the topic. Yeah, then there's a lot of rodent systems that are used to study stress transmission on. I also choose a study of stress transmission in mice here. So this is a study done by a neuroscience lab, which are studying the impact of stress actually in neural circuits. So in particular, they were interested in neurons that control the release of hormones via the HPA axis. So they fire to release corticotropin releasing hormone, the CRH, upon experience in a stressor. So the setup was that they have 3 mice housed together, and then in separation, one of them receives a mild electric shock, a foot shock. And then, so this is the synaptic response to the stressor of the subject. And then the subject is brought back together with one of the partners. And then the partner 1 responds even more strongly upon encounter with the stressed mouse, but it's like giving the same pattern as the first mouse, the subject gave to the stressor. And then the first partner is put back together with the second partner. And so, yeah, partner 2, who never even encountered the directly stressed mouse anymore, is still, yeah, showing a very similar stress response profile. This study also gave support for the idea that alarm pheromones that are secreted by stressed individuals are involved in this process, because the time the animals spent with anogenital sniffing was much increased when they were put together with a stressed animal, and also when the animals were just presented with a swab with the scent of a stressed individual, they would also could respond with a stress response themselves. So I don't know if this is something that's relevant in all systems, because in birds, for example, it was long doubted that olfactory systems even play a role at all. I don't think it's that true to that extent, but yeah, there might also be definitely other systems involved in the transmission. So that's also something where there's a lot of open question. So yeah, if you by now you started worrying about your stressed friends and colleagues transmitting stress to you, maybe you should be, but there are also some good news. So not all interactions with stressed individuals have always to result in stress being transmitted, obviously. Alternatively to stress being transmitted, social buffering can also occur, where social bonds can actually facilitate the downregulation of the stress response of another individual. So this is again more likely to occur between individuals with strong bonds, but not exclusively. And so the individual that is buffering the stress of the partner can experience an increase in their own stress levels in return after the buffering, which was shown in rats, for example. And sometimes people also refer to this process as co-regulation, so hormonal or emotional co-regulation, because yeah, if this goes both ways, it's actually rather a synchronization process than a one-way transmission or contagion, which I yeah, think is quite interesting and something to think about. But yeah, as in another study on group living fish, it was also shown that the buffering individual received fewer positive social interactions after it was buffering a partner. So while social buffering can provide protective effects on stress responses and possibly the subsequent negative health consequences, it might also reduce the buffering capacity of individuals and the group for the future. So if the social connectivity of the group actually becomes reduced after social buffering events, this might then lead to other interesting changes of the dynamics in the group. So the reduced connectivity could then either prevent social buffering in a future stress encounter, or it might also block stress transmission in a future event because of a lacking transmission pathway, basically, or the lack of change in connectivity might then represent a stress to the group itself. So based on these examples, we can expect quite complex dynamics around these physiological interactions, as they might also strongly affect the structure of groups. And this is also a field where so far only very little research has been done. And yeah, for now we're just trying to puzzle together the bits and pieces of information that we have already. But yeah, I think from the evidence that we have that this could be a really important process in animal collectives, which is why we just published this review and perspective article on the topic together with my collaborators that came out earlier this year, where we discussed possible functions and consequences of stress transmission in animal collectives. So the general idea is that if an individual in a group encounters a stressor and triggers a stress response, it could either directly signal the presence of a stressor, maybe by an alarm call or pheromone, or the activation of the stress system could also be perceived by others as an indirect cue, so basically public information, and then spread via social stress transmission as a form of social contagion. So examples could obviously be directly signaling a predator encounter to others that might then be able to quickly respond and trigger stress response before having encountered the stressor themselves. But as a more indirect cue where others are not trying to signal, we could think, for example, of just of a dog going to the vet that's generally fine with it, but then gets really agitated when it enters the vet office because it actually perceives the scent of the other stressed animals that have visited there. Or if we translate this to the wild, we can, for example, think of individuals prospecting for a new habitat, a breeding area, or a social group to join, and then the stress levels of the others there might serve as a cue about food condition or predator abundance. So yeah, just put this more of a positive, I think. I don't think it has exactly been tested in this form, but I hope that someone will. Yeah, the other option is that the stress gets buffered by others in the group. But yeah, at the moment, we don't really have a good understanding of what conditions stress might get buffered or be transmitted, and whether strong social bonds or stronger cohesion in the group makes it more likely for one over the other to occur. And yeah, whether stress needs to overcome an activation threshold or so to actually be transmitted with others responding strongly enough. But at the end, if the stress levels in the group become amplified, this could impact social interactions, the collective performance, could impact things like group foraging, colonial breeding, predator avoidance, or flocking behavior. And this process we can then obviously expect to feed back on the individual, so it might lead to a different outcome if then an individual encounters another stressor in the future, because it might have a different activation threshold. It might change the likelihood of encountering a stressor because it might have different activity levels or movement patterns. So generally, if the stress response of one or few individuals can spread and affect others that have not even experienced the stressor themselves, could amplify the effects of stressors that individuals may encounter and increase the frequency and duration of the stress response, which could then really change this balance between the costs and benefits. So for this, it would be important to better understand the dynamics of when stress transmission might take place. So one of the big questions here methodologically is how we can study stress transmission. I think one of the reasons why this has not been studied more, and that it's really challenging to study, a major problem here and generally in stress studies being how we can measure or manipulate stress without acting as a stressor while doing so. And this problem really doesn't become easier when we're looking at a group of animals. So generally, I also think we can say that there's not really one reliable marker for the stress that an individual is experiencing. So many studies rely on endocrinological methods, mostly measuring glucocorticoids from blood, hair, feathers, or fecal samples. But it's not always as straightforward as we sometimes like to make it seem, mainly because there's a lot of individual variation and fluctuation. It can be difficult to compare between individuals, between different time points, and to find the right temporal resolution for what we're trying to measure. And there are a multitude of other physiological parameters we can take from samples, like oxidative stress, DNA damage, so on. We can also take other physiological measurements that can happen non-invasively, like thermal imaging techniques, where temperature increase around the eye region was often shown to correlate with the neuroendocrine stress response. Heart rate transponders, like these implantable ones or external transmitters, can be really valuable for field studies, but also in captivity, because we can get very fine-scale readouts and even measure some of the faster responses from the sympathetic nervous system. Yeah, and last but not least, we can try capturing behavioral responses and adjustments by different camera tracking methods. So one approach that was developed from people in my lab, Adriana knows quite a lot about this, and I've also used this quite successfully in my experiments. That is the barcode tracking method. Every bird wears a little backpack with a unique QR barcode. So it's also been used in other animals, I think even in ants. And then we can capture from above with just a cheap Raspberry Pi camera mounted over the perches, for example, and we can extract the location and orientation of every bird at every second. Meanwhile, we have even developed a markerless tracking approach with a deep learning-based method that allows the individual identification of birds. So we were also able to apply this to the several finches in the cages, but it can also be used for birds in the wild, which I think can open up a lot of new opportunities. Yeah, so this is obviously by no means a complete comprehensive guide on methods, but just a few thoughts and ideas really of mainly some methods that I've looked into myself. And I also included this today because I find that this is something that people ask about very often. Yeah, and measuring stress alongside with how can we manipulate stress. Yeah, which is something that we might also sometimes want to do for experimental studies. So to manipulate stress, I think there are two main categories of approaches that I'm aware of. And I'm also discussing this now because it's relevant in the experimental work that I'm presenting later, where I work with both of these approaches actually. So one is a pharmaceutical manipulation, often consists of administering glucocorticoids either orally or via various types of implants to simulate elevations of these hormones, as we might find during or after stress response. So some of the problems associated with giving glucocorticoids is that we're giving often high doses of this general metabolic hormone. It's not entirely clear, I would say, what all effects we might cause by that, how much this really resembles what would occur in a stress response. It obviously also involves many other hormones and nervous responses, and particularly higher doses that are often given in such studies then also lead to this negative feedback of the stress response system that then down-regulates the response and leads to a weakened stress response subsequently. But one point is I see as a big advantage with this method, especially thinking of stress transmission, where one will study multiple animals in a group, is that it can be done with a one-off manipulation in case of the implants, possibly even without human intervention via food or water. And yeah, so this might sometimes make this a bit more feasible. And another approach would be to try and mimic more of a natural stress encounter. So there's, for example, a protocol that was developed to induce chronic psychological stress in birds by exposing animals to changing stressors at different times of the day. So the animal will not habituate to it, and the unpredictability might also cause an anticipatory stress. And so the different stressors can consist of things like restraining them in a bag, exposing them to loud music or human voices, or disturbing their cages by rolling or tapping them for 30 minutes or so at a time. Yeah, but obviously many things conserve as stress to animals, and other manipulations might involve social isolation, food restriction, or other physical stressors like temperature manipulation, so on. And sometimes people might just get very creative and come up with robotic lions or yeah, some devices that make it not necessary to interact with the animals while applying some stresses. So the methods are endless, I would say, but yeah, not always easy to predict really what result or simulate the result we want to get. Yeah, so to wrap up the methods part, I'm now moving on to the next part where I want to introduce the study species that I've been working with for a few years now, that are zebra finches. So zebra finches are among the best known colonial breeders. They are popular model species that people really love to study in the lab. But to really understand the social processes and social structure, it's always good to take the ecological context into account. So I studied them in the wild, in the Australian outback, trying to understand a bit how being social helps these tiny birds survive in the very unpredictable and harsh environment. And so this is also, of course, now where I draw some ideas from when I think about how stress might disrupt the social group. So because of the ecology, zebra finches are really not like your typical European songbird. They breed opportunistically, which means that the breeding is not bound to a fixed seasonality, but they breed whenever environmental conditions are good. So when rainfall set in, that can also be very local, makes the grass seeds grow, which they then use to feed their offspring. And so for them, staying in social groups might also help them to be more ready for breeding and then quickly form a breeding colony when these rainfalls set in. So the study site in New South Wales in Australia, where they set up these artificial nest box colonies, arranged like breeding colonies, and they very readily actually accept these nest boxes. Attacked about 1,000 zebra finches with these passive integrated transponders that can just be injected under the skin, and then allowed me to track the activity. So I used RFID readers with antennas to track the breeding activity in the nest boxes, and the visits also to this feeding station. So one thing that I was interested in was the breeding synchrony in the colonies, because the reproductive timing is actually so variable and can also go over extended periods with multiple broods if conditions remain to be good over longer periods. Yeah, but what I found in these colonies is that they actually don't exhibit a high level of breeding synchrony. So in a colony, I would usually find nests at all different stages. So they are nest building, incubating, or rearing chicks next to each other at the same time. However, from my observations, I sometimes had the impression that there were smaller clusters of a few synchronized nests close to each other. So I did an experiment to test whether the birds are actually actively synchronizing the breeding with some of the neighbors. So for this, I placed an empty nest box next to an existing box at each of the different stages. And I predicted that in order to synchronize, zebra finches would be more likely to initiate breeding next to a stage at a similar stage. So that would be the early nest building stage. And what I found was that they were indeed much more likely to start breeding in a box neighboring the stimulus box with a pair at the nest building stage. There were also some cases where the stimulus nest was then abandoned during the trial, in which case it was also less likely that birds initiated breeding in the experimental box next to it. So I think this makes it even more clear that the neighboring nest acts as a social stimulus. So it seems that the zebra finches actively choose neighbors to synchronize their breeding with. And one advantage of this could be that the chicks will fledge together, and chicks from different nests and also stay together in creches after fledging, which might again help them to survive better. And so after I found this result, I was wondering if the synchrony between certain neighbors is linked to the social structure of the colony, and if these associations then persistent so that we would find the same in other behavioral contexts, or if those are just purely opportunistic short-term associations for breeding. So we looked at the social networks outside of the colonies and also outside of the breeding period to understand who's foraging together and how they are associating in the feeders that we put up in the surrounding area outside of the colonies. So I looked at the social network of the breeding pairs in 3 different periods. First, a longer breeding period in the first year. We looked at the synchrony of the nests in the colony, and I found that the neighboring pairs with higher breeding synchrony have stronger social bonds than expected by chance in the feeding context than the less synchronized breeders. And this was true in the first year, but also after 8 months with no breeding activity. In the next year, the social bonds of those birds that have bred more synchronously in the first year were still much stronger. So the birds that bred together synchronously also feed together outside of the colonies in what seems to be quite persistent associations. So I also checked again after the end of the next breeding period in the second year, but at that point the original sample was strongly reduced because the small birds sometimes just evaporate in the outback. So I could not really say for sure because the sample size was too low, but the effect size was actually still very similar as in the other periods. So it could well be that they maintain the bonds over the breeding period or over multiple breeding periods. So the breeding dynamics in the colony are really strongly linked to the social structure, even outside of the colonies. And now, even additionally to this dynamics in the colonies, who individuals associate with might also be shaped by individual traits, like the social phenotype or physiological state. So because I was interested in this, we know that social behavior can already be shaped very early in the life. We had some prediction from an experiment that was actually done before that work on captive birds, where stress exposure during the nesting phase shaped how the birds socialize later in life. Now we wanted to test this prediction in the wild. So we used sibling competition as a natural stressor. So I manipulated fruit sizes, 6 chicks or 2 chicks in a nest, which is both in the range of natural zebra-finch fruit sizes. And after this, represented higher levels of stress. And after these birds then fledged from the nest, we could follow who they were foraging with and associating with again in these feeding stations. And so what we found was that the chicks that were exposed to high levels of sibling competition foraged with more individuals. They were less choosy in their associations, and they had more central positions in their social network, all of which might had prepared them for becoming independent early more easily after growing up in a potentially difficult environment that it might be good to escape from possibly. So and these are the same results that were found in the lab study before, prior to this work, where the chicks were fed with corticosterone instead of the sibling competition as a stressor. So we can see that in these examples how these processes are strongly interlinked, and this idea is also summarized in this review that came out of our lab last year on the feedback between the individuals and the society, but also how the social structure and the processes emerging from it in turn reflect back on the individual. So for example, the social phenotype of the individual is already shaped in the nest, affects how it socializes, will ultimately scale up to the social structure in the breeding colony, who's breeding next to whom, who are they foraging with, and this influences all social and ecological processes and consequence, which in turn will impact on the individual state, for example, by affecting how much competition they experience, how many siblings they have, how much food is available, and so on. So now linking this back to the questions that I introduced at the beginning, how social groups deal with stress and the dynamics of stress transmission, we can really see the same idea, the same pattern, same feedback loop repeated here. And now to get some answers to test some of my ideas and hypotheses of what impact stress can have in the social environment, I took this back to the lab because it allowed me more fine-scale tracking of the individuals that is useful to capture fine behavioral nuances. And so I designed an experiment using captive zebra finches for this. So I started with 96 birds, 48 pairs. For the treatment phase, I split them up in 16 cages, basically simulating sub-colonies of 3 pairs each. And so I had 3 different types of treatment cages. These are no birds in the cage received the treatment, that is the control cages, or 2 of the birds received the stress treatment. Partners always got the same treatment, or many of the birds were stressed. It was 2 out of the 3 pairs. So this treatment phase was repeated in 3 rounds, where every bird was once stress treated and served with a control in the others. For the stress treatment, as I discussed before, because there are pros and cons for the different methods, and I wasn't sure what would be the most appropriate method for stress transmission, I did use 2 different approaches on half of the birds each. So one was a hormone implant releasing corticosterone over 14 days while self-dissolving. So this was a one-time procedure and they did not have to handle the birds for the rest of the experiment. As controls, I had placebo implanted birds to control for the manipulation. And then secondly, I also used this behavioral stress protocol to induce chronic psychological stress to mimic more of a natural stress exposure. So here the birds were caught every night at unpredictable times. So I would randomly show up at 8 p.m. or 3 a.m. to catch the treatment birds in the dark to not disturb the other birds. And then for 30 minutes, I exposed them to changing behavioral psychological stresses, which was either just restraint or cage disturbance, or making them listen to loud, angry German rock music. So this was the treatment. Then this is a top view of one of the experimental cages. So the birds were constantly tracked with the unique QR barcode backpacks with 64 cameras in total, each taking 1 picture per second of the position and orientation of the birds. So I think I got something like 116 million bird detections from that, which I then used to measure the movement across the cage and the strength of the social relationships. I measured the body condition with feather samples to measure cord levels, and I monitored the breeding activity using RFID readers and eye buttons, plotting temperature in the nest. I cannot talk about all of this today, but I will show you a few selected results now. So I will, I will show you the results in the form of model predictions, testing the effects of treatment conditions on an activity measure, group cohesion, and reproductive parameter. And in this result, I will always first on the left be showing the direct effect of the stress manipulation, because I did not always have a clear prediction how, for example, movement would change under stress. So first tried to get the baseline for the effect. We expected the strongest difference comparing only non-stress exposed and the most stress exposed birds. And on the right, I will then show the effect of the stress composition on the environment by using the number of stressed birds in each group as a predictor, which, while obviously considering what treatments the birds have received. So if the unmanipulated birds in these groups are affected by the stress in the environment, this could then be considered an effect of stress transmission. And I expected that the effect size might then depend on the stress composition of the group, so whether 1 or 2/3 of the group are stressed. So first I was interested in how birds move when they're stressed. So one of the movement variables I measured was the percentage of time birds were changing between areas of the cage within an hour, so basically hopping from one perch to the other. And I found that the stress-treated birds in yellow showed decreased movement compared to the respective controls in blue. So on the left, we have the behavioral treated birds, and stressed, non-stressed, and on the right, the implant-treated birds with the corticosterone implant and the control. And so I found that the stress-treated birds in yellow had an average, you can predict 6% fewer area changes in the stressed birds. And also there was an effect of a difference in the treatment. So there was additionally about 5% less movement in the cord implanted birds compared to the behaviorally treated birds. If we have a look at what happens in response to the stress in the social environment, where there are more or less stressed birds in the cage, so there was an interaction between the number of stressed birds and the environment and the treatment that birds received. So birds that were already stressed themselves responded much more strongly to the stress in the environment, were reducing the movement much more than control birds. And implanted birds, also again had somewhat lower activity levels than behavioral stress birds. So here, I think it's not absolutely surprising, but definitely interesting that if you're stressed already, you might be perceiving the environment differently than the other birds. Next, I had a look at a measure of group cohesion. So I measured the distance between basically every bird and all of its cage mates during the treatment, and we can see that especially in the behavioral stress group, so there was big difference between the distance between the individuals was much increased in the stress treatment. In the implanted birds, this could, the fact that the control implant also showed the same higher distance between individuals could also be an evidence that maybe the implantation of the control implant, of the placebo implant, might also have had an effect here. If we look at the social environment, the number of stressed birds in the cage increased the distance between individuals across all the birds. So there was over a centimeter additional distance between individuals, basically per stressed bird in the cage. And these were rather small cages, so this is already a bit of a distance when we have more stressed birds in the group. Last but not least, I looked at the synchrony of reproductive timing within the cages. So the unit is now the cage, not the individual. They use the difference in days between when the first and last clutch in a cage were started as the response. So now we have the cages without stressed birds on the left of the x-axis, but the maximum difference between clutch start is on average about 2.5 days, and cages with stressed birds on the right. And we see that the difference in the behavioral stress group especially become larger. The effect was not significant here, but also this is obviously a much smaller sample size here, because now the cage is the unit. But I think that there could be in larger groups or with more groups that could certainly be an effect on the reproductive synchrony. I also started to see some results in the pair relationship between co-treated pairs, increased the relationships actually where behavioral stress birds, which is I think more of a psychological stressor, seem to be more disturbed in the relationship and in the synchrony, which could indicate that the stress could strongly impact on some of these dynamics. But I don't find all of the results super easy to interpret, but I do see some overall patterns. But as always, it's a bit of noisy data, and we see different impacts on different variables. So to wrap this up a little bit, I think that stress can obviously have effect on many parameters, on the individual's movement, social behavior, reproduction. This is not necessarily new or surprising. I mean, this has been studied on individuals for years, but the stress of other group members in the social environment, I think, can also really affect individuals that were not exposed to a stressor themselves. There's a lot of individual variation in the responses, so this is something, that sometimes creates noise in the data that is hard to interpret, because there's just so much variation. Actually, the psychologists, when they analyze this data, they upfront basically split the test subjects in responders and non-responders, and then only measure the response, the height of the response of individuals that are responsive to transmitted stress in first place. So basically that would be filtering out all the outliers of my data that I don't want to be there. But I think we just have to accept that there's a lot of variation, and try to find some interesting patterns in the noise if there are any. But I certainly think that there are some effects, but there's also differences between the different stress manipulations, and there's differences between the different behavioral contexts that we're measuring. And I think this also makes sense biologically. It's just, probably requires a bit more work to tease this apart on all layers. But generally, I would say that both stress and stress transmission can affect social groups, but we do have a lot of remaining open questions. So in my next steps, I'm actually trying to take this a little bit back to the wild. This was some lab experiments to have a bit of a proof of concept and showing a bit what's happening in birds in general. But now I actually want to see what's happening in social groups in the wild and the role of stress there. And so one collaboration I'm having is in the Siberian Shale long-term project with Michael Frisser, where this family living core weights that can be attracted to the feeding stations, where we can observe the behavior. There's a lot of differences in aggression between the group, between the dominant breeding pair and then kin and non-kin members. Yeah, that some of them have higher levels of aggression, and we can collect the feathers and yeah, look at the stress levels over certain periods and also over the early life periods. I think it's a very suitable system to look at some of these questions that I'm interested in. And then also I want to look at the vulturine guinea fowl in a continued collaboration with Damien Perrine. So there's all these big blue chickens in Kenya that probably shouldn't call them that, but yeah, they have they're very social terrestrial birds with which we can track with these GPS trackers, and also many of them have heart rate loggers and accelerometers, so we can really look at the dynamics in the social group and the fast heart rate responses, how they're responding in the group to different social factors and environmental factors around them. So yeah, I'm very excited to look a bit more into explore these questions further. Yeah, many thanks for listening today, and everybody who was involved in all this ongoing and past work. I also, just while working on this, I started to put together a little slide with open questions, because I always came up with things that need to be, I think, should be studied. Also maybe just to put it up for, I don't know if people have any thoughts on this. I'm very happy to discuss any of this. So yeah, what defines whether stress is suffered or transmitted by others? Are those even different processes? That was what I touched on briefly with this co-regulation. So it's always like a both-way process where it's just trying to reach synchronization of these distinct processes. What makes one happen over the other? What is even the value of achieving synchrony? What is the role of individual differences? I think that maybe also the level of individual differences in the response might also help social groups to buffer a bit, maybe from the effect of overreaction. Maybe it's not even good for everyone to be stressed equally in the group at the same time. Yeah, what are the mechanisms involved in transmitting stress? So I mentioned a bit that we have some evidence for olfactory cues. There might obviously be acoustic signals. I think it's probably a multimodal process. I think it's possible via various channels, but we don't really know it that well. In what context does stress transmission play a role in the wild? And when does stress transmission in groups lead to negative fitness consequences, which is something that's really an ongoing debate, even in individuals. But yeah, I think for me, even more interesting in groups. Okay, I'm gonna finish now with my part, but yeah, I'm very, very curious to hear questions and comments from everyone.
Thank you, Hanja, for such a great talk. We're gonna open it. We're gonna open it for questions. So as a reminder, just type in question mark, and I will call your name and relay the question. Also, Lauren, if you have any question on YouTube, please keep an eye on it. Well, we get questions coming. I'm gonna get started with one that I have myself, taking advantage of being the host. Have you, or do you know if there is any mechanism of self-regulation of stress levels in individuals? Like, I mean, we do it ourselves somehow, and I don't know if that's sort of the same in animals. So
Sorry, if the self-regulation of the stress response, like kind of self-calming behavior or something. Interesting. Hadn't actually thought of that. I mean, I mentioned there's probably something that's been studied in humans at least. I actually haven't thought about this at all in other animals. I don't know who asked this question. I would be curious if there's any answer to it.
No, it's great. It was just curious because I mean, in a group, we often get stressed, right? And stress is socially transmitted, but at the same time, we try to regulate it and try not to get as stressed as everyone else is in the group. So I was wondering, maybe, I don't know, if we require a cognitive process to sort of achieve that kind of regulation, or if you can actually see a change in stress levels throughout the transmission. Yeah,
I don't know, but would also be interesting if there might be a thing like, for example, like diseased animals that sometimes like try to isolate themselves from the group to not transmit it. So there could be a thing of stressed individuals trying to isolate from the group to not transmit stress to others. Basically taking a time out, a little bit of me time. I don't know of any documentation of this, but it's an interesting thought.
yeah, interesting. Thank you. Okay, so we have a question from Julie.
Hi, can you hear me?
Yes.
Okay, thank you, Hanja, for the talk. It was really, really interesting. I'm working with stress in spider monkeys, and so the question goes a little bit towards looking at your results. I have the feeling that the physiological approach was a little less pronounced than the behavioral approach. And in primates, primates don't really use a lot of like olfactory or cues for knowing the state of the other individuals, and I don't know how it is like in birds, whether there is some behavior that indicates whether the other animals are stressed. Like in primates, you would see certain self-directed behaviors, for example, to indicate that an individual is anxious or other types of behaviors, you know, like it's more visual clues that indicate the stress level. So maybe is there something, did you look into like visual cues for the other ones to see whether or not the stressed animals are stressed?
Yeah, it's a good question. I think what would be really interesting there, what I was actually a bit curious about, I haven't really implemented it yet, but it would be posture tracking, where I think if we could like, pick up really fine scale nuances. On a broader behavioral level, I mean, there is some documentation of some stress behavior. For example, that would be perch hopping. So that was the first movement variable where I showed where I found actually a decrease in the movement activity. So this is something that's described commonly in birds. Generally, birds are very good at hiding when they're not well. So also when they're sick or injured, or so they're really. I think it takes much longer than in other animals for them to show. So I could also imagine that they may be not showing stress so strongly. If you have pet birds, I mean, there's things like, I don't know, when they start plugging their feathers and so on. It's not, I think it's something that you probably need to go from species to species. So I didn't wanna like make too strong assumptions from behaviors that are described in one study that I necessarily expect this behavior. This was rather why I just wanted to look what's happening from my stress treatment, and then make my assumptions based on that. But yeah, I don't think that there's like one behavior that we can say this is 100% foolproof, this animal is stressed, because I think it's just too hard to interpret, and could probably all these other causes that an animal is moving somewhere. So, but yeah, sometimes avoidance behavior, obviously, of some stressors. So I think it depends a bit on the specifics. Did this kind of answer a bit? Or aggressive behavior also could be triggered, I think, with stress,
But so you think it's more like behavioral indicators that transmit the stress rather than...
Yeah, what's transmitting it? I think that it could be multiple things. I was also like interested in doing maybe experiments with the acoustic. I mean, when you have the zebra finches, they're just vocalizing the whole time. So obviously acoustic cues could be a big thing there. I don't think that stress pheromones play as big of a role as in mice, for example, but we cannot rule it out completely. I would, I would think it would be interesting to do a study where you would, just basically take away different sensory channels and just test them one by one and in combinations. I could really imagine that it's not just one thing that gives it away. And
Okay.
especially in pairs, it could also be different again. Like if you're building a nest together, you might pick up other cues than when you're just a neighbor that you're not trying to build a nest with right now. Yeah.
Okay. Okay, thank you very much.
Thank you.
Okay, before we move on to the next question, Julie, could you please state your full name and your affiliation? And I remind everyone to also do it, as we are trying to build a community, and some of us are old folks here, but we love to hear from the new ones. Julie?
Oh, sorry. Yeah, I'm Juliane Dam, and I'm at the University of Veracruz in Mexico. I'm working with Filippo Aureli.
Great, welcome.
Thank you.
Our next question comes from Maren.
Hello, I'm Maren from the University of Derby. And my question is sort of similar to that of Julie, and also essentially your third point of your open questions. So how do animals know that somebody else is stressed, and is it the hormones of the others? Is it and how would they pick it up? So for that they would need pheromones. Is it the behavior? And it's it also reminds me of this saying, do you cry because you're sad, or are you sad because you're crying? And it's sort of similar with the stress. Are you stressed because your cortisol or whatever goes up, or does your cortisol level go up because you're stressed? And I have very little understanding of these endocrinological underpinnings and how it all works. And I really found it fascinating that you had these differences between the behaviorally stressed animals and the cortisol stressed animals, and a little bit more insight on how this all comes together, if you can.
I think we definitely need a lot of more research, and I think a lot of the work that has been done there was quite I think heavy on the rodent systems, which I think gives I mean the study represented at the start really showing like really how this I think definitely for the are you sad because you cry question, we can say that I think it is I mean it starts in the brain, the stress response starts in the brain, and I think the hormonal neuronal output that we see as a consequence after the stress, having perceived something and triggering this, what pathways it takes. I mean this is a bit what I just said to the previous question. It's difficult. I don't really have answers for it at the moment. I think that it also doesn't necessarily just one thing. Also, I mean, in the human studies, we've seen like a lot of these things where really also these subtle unconscious things are perceived, like really changes in heart rate, where you synchronize the heart rate with your partner. I mean, that's not something that you're actively perceiving. I'm never like sitting, unless somebody's like really crazy out of breath or so, I would never like think, oh, this person has a nice heart rate, let me match that. So this is, I think, really also a lot of things that where we just have to take more physiological measurements as well to look into this process. As I would also be very interested to combine such studies with heart rate monitors. My problem a bit why I didn't have more physiological measurements is because it turned out that several finches are very small. And it's just very hard to take physiological measurements, especially by keeping them alive. Yeah, I did get a little bit of pilot data, but yeah, not really sufficient. But yeah, it's just something to build upon.
Great, thank you. Before moving into the next question, I would like to remind people watching us on YouTube that you can type in your questions and they will be relayed to me by one of our colleagues. Our next question comes from Eduardo.
Thank you. I'm Eduardo Fernandez Duque from Yale University in the US. And I'm one of the old folks that Adriana mentioned. And I'm one, like Maren, somebody who listens to the endocrinology side of the story, but quite a bit goes over my head. So I don't know if it's a question, but I really appreciate it. Really enjoy your talk. And I really appreciate it that you finished, and we still have it there, this slide, inviting us, that you finish with questions, which I think it's usually how the things really go. Whether we like it or not, we finish with questions. Seldom we finish with conclusive answers. Going back to the primates, and then trying to relay my comment on the primates, which is my system of study, then going to the specific question for you, Hanja. I keep telling the students that we start making plans for research, thinking about what we should measure. And then we settle for what we can measure. And the mistake we keep doing is assuming that we measure what we should. At the end of the day, we measure what we could. And the reason we have more information linking or trying to link stress in primates with behavior instead of physiology is because we can hardly do anything with physiology in the field. And the best we do is just collect some poop and then hand wave about whatever it is we measure in poop says about stress. So I think that should be a little bit of, for you, it's fun. I think it's fantastic because I'm sure you've learned so much of this moving through so many different systems, all of them with their own challenges and benefits. Help me out with how you define stress. And I know this is like asking you, we were talking when we met, pair bonding happiness, but how should we be thinking these days? Because my understanding is that, and what I tell the students, cortisol is not a stress hormone. Forget it, drop it. But anyway, we keep falling into that. And we fall into that because that's the only thing we can measure, somebody says. So, and then there's no clear answer, I know that, but help me out with, if I go back tomorrow to the students, how should I start talking about what stress is in the context of behavioral ecology of non-human animals?
Yeah, I mean, this is all very, very great points that you're making here. I mean, first, I fully agree that at the end we also have to be obviously super opportunistic about what we can measure, especially when we already have a fixed study species. I mean, ideally you go out there and look for study species that fits your question, and sometimes it might not be the species you have in front of you, which is happening to me a bit with the zebra finches. I just realized, it's very limiting in the methods that you can apply in some ways, but then they're good for other things. So I think we need to combine multiple methods and probably multiple systems to get put together these puzzle pieces and get different sides of the coin, maybe. What is stress? I mean, I think telling students that corticosterone is not a stress hormone is already a great job. Just being aware of that, I think it's number one. That we just treat it a bit carefully when we look at it, that we know it doesn't necessarily mean one thing. I don't think there's one good reliable stress marker on what an animal is perceiving. Generally, I define stress as having a physiological stress response. However, we can show that, which is often just we're estimating it by the cortisol level, ideally not change from before to after. Because sometimes it's just the best we can do. Ideally, we integrate multiple approaches. I think that's the best. Validating the method that we have. I mean, if I'm stressing an animal, I measured it before and after, I see a certain increase. I assume that it had a stress response meanwhile. yeah, we just have to go with what we know also at some point. Yeah, there's also I read a study in humans a while ago where it turned out that yeah, what people report as feeling stressed does not at all like match the biomedical readout of who's stressed. So this is also like the psychological level, the emotional level, which is like where I'm not even touching on when I'm working with birds, but then again, yeah, I do think that animals have the personalities, and if you ever had any pet of any type, I think you probably would also say that they can be very emotional. Yeah, I try to not go there with my research, but yeah, there's another level where I cannot really say how they're feeling stressed. Yeah, it's just we have to. I think we just have to do the best that we can with the tools that we have. Yeah.
Thanks. I think you're on the right path, because I think it's going to be so enriching that you're trying to learn about this on different systems. That's going to be, I'm sure, very powerful. Thank you. So thank you so much.
Thank you, Eduardo. And we have another question coming from Luca.
Hello, can you hear me?
Yes.
Yeah, I'm Luca. I'm a PhD student at the University of Exeter. So I was wondering, first of all, thank you very much for the very interesting talk. I really enjoyed it. So I was wondering if social buffering and social transmission of stress are sort of complementary processes or two sides of one coin. I was wondering, what do you think, under what circumstances should we expect either one of the processes to be more relevant, or when should buffering occur and when should transmission occur?
Yeah, I really don't know. This is why I put it out as a question there for you guys. No. Yeah, it's something that I really, I mean, I think that sometimes stress transmission has this informational value, where yeah, it's warning others in some way. I think it might also sometimes really depend on the state of the receiving individuals. So there was in some of the results where it showed that the birds that were already stressed seem to be more receptive to stress transmission. So maybe if you already have a certain level of some type of agitation, maybe already a certain level of stress hormones, whatever, you might be, and then you encounter an individual with a stress response. This might be basically reassuring you in your stress, and could then lead to stress transmission, is what I could imagine, versus when you are actually feeling very safe, very, yeah, not you don't think there's a reason to be stressed. It might be buffering. So in a group, I could imagine that if many individuals are already stressed, that the stress would rather spread further. Maybe if then just one individual comes in and the others are not stressed, this could be rather buffering the stress of the other. But I don't have very good data to make the statements. I mean, I do have like a little bit of evidence here and there where I'm making up this hypothesis, but it's really the best I can say about it at the moment from what I've been thinking about it. That I think could be some of the dynamics that are involved in the groups. But yeah, I don't think anybody has like, I don't know of any studies where we could like with some experimental paradigms switch between stress transmission and buffering. I don't necessarily think that it's always a process in both directions, but sometimes, yeah, it's, I don't know.
Great, thank you.
Thank you, Luca, and welcome to the community. Next question comes from Veronica.
Hello, everyone. I'm Veronica Beek. I'm from University of Vienna. Nice to see you again, Hanja. It's been a while. Thank you for that very interesting talk. I'm studying elephants at the moment, which I know is a very different system. But here I can see a lot of consolation behavior when one animal is stressed. by an external stressor or also social stressor, that when they have strong social bonds, that the other individuals would try to calm them after stressor. And I was wondering if something like this is happening in Sept Finches too, that it's not only social buffering because there are others there, but there's also some kind of active consolation or mitigating behavior.
Yeah, it's a good question. I'm not entirely sure. I could definitely see it within the pairs for sure. I mean, they do sometimes screen each other. I have not actually measured whether this would be happening more after when one is stressed, especially because in my setup now I stress the pairs together. So maybe also that's probably would be an interesting question if one of them was stressed or so, to see if then the partner would receive more consolidating behavior, whether this is calming them. I know that some studies on that have been done in a few other species for sure. Don't make me name them. I think there has been some description of consolidating behavior in birds in general. Yeah, in the Sept Finches, I have not exactly looked at this, mainly because I've been processing now rather large scale data rather than like very fine scale, where I've been like I haven't been analyzing like every behavior from video cameras, but rather look through these giant data frames where I'm using automated data extraction, and then I can look easily at who's sitting next to whom and where are they moving at every second, but it's hard to say whether they're consolidating each other just because they're sitting next to each other. It's also a bit a question, what would be the exact definition of it? Do you have a definition for it in elephants?
Yeah, it depends on whether they do it more after a stressor than compared just doing normal social situations. So
Yeah. Also, I think in my setup now in my study, also because I introduced more of a chronic stressor, I think that was probably something what you maybe need to have more acute short stressors to get the response from it. Otherwise, it's probably hard to align it on a time scale. But yeah, it's interesting. Maybe something to consider for another study. Thanks.
Thank you.
Thank you, Veronica, and welcome to the FINE community. Our next question comes from Lauren.
Hello, my name is Lauren Hayes. I'm University of Tennessee at Chattanooga, and I study social rodents. Thanks for that really interesting talk. And I'm almost going to apologize in advance because I'm not sure my question is going to be clear, and I may offend Eduardo for asking a question about primates that seems dumb. So to what do we know to what extent eavesdropping occurs and how that may play a role in sort of how, like, for example, a primate group may notice that there's social transmission of stress and there's things going on and now's the time to attack? Do we know anything about this? And is this even interesting, I guess, is the question.
I asking it about primates specifically, because like I will not be able to answer that. Maybe Eduardo is. I don't know. In Sept Finches, they're very peaceful individuals that will not do any warfare against each other. So generally, I mean, eavesdropping, maybe in other contexts, is definitely something that's happening in birds, but maybe rather where somebody's catching the food or something, examples like that. Yeah, in Sept Finches, I cannot think of any example in this context. But yeah, others exploiting the situation. Yeah, super interesting. Yeah, it could be a thing. I mean, that maybe a bit in terms of what I was suggesting at some point. That in the context of, for example, prospecting different habitats, you might get an idea also of other social groups when you're exploring other social groups, and you might perceive the stress levels, whatever you do with that information. Then if you plan to plan a little warfare against them or to exploit the food caches or or steal the roosting tree. But yeah, could be something that could be used to your advantage potentially. I cannot really think of an example where I think this happening in birds, but could definitely be a thing in some system.
How are you thinking of eavesdropping, Lauren? Can you help me out with elaborating? What were you thinking?
I guess you're just paying attention to what's going on in the other group, and I guess you have the ability to assess the situation, to say this group is experiencing a lot of distress. This would be a great time to try and take over territory. I don't know if that's plausible or not. I study rodents, so it's. Could another?
No, I was thinking. I mean, go ahead with.
No, please go ahead. Go ahead.
No. Ask her. She's the.
Could another context be that a predator could sort of cue in on this, and
it would be interspecies stress transmission. Would also be interesting. I don't know. I cannot also not really think of a scenario where a predator could maybe on alarm calls. I think eavesdropping on alarm calls of others is definitely a thing. In mixed species flocks, for example, some birds exploit the alarm calls of others. I guess that could be considered in this direction. Yeah.
I was thinking about the same, but then that's I go back to my question, which wasn't a question, was more of a reflection, that I can totally imagine some jays calling like crazy in the forest, and different groups of monkeys, or actually maybe monkeys giving alarm calls and groups nearby noticing them. But is that going to be stress? Because if stress is anything, then it's nothing, right? So if any input that I take in that has the potential to elicit a physiological response along the lines of how Hanja described it in the first few slides, then what do we do? Yes, I can totally see eavesdropping, a small groups of primates giving alarm calls of some kind that the neighbors may associate with a potential risk. That's as far as.
It sort of happens that way in rodents as well, Lauren. At least in marmots, I've seen some neighboring groups that they pay attention. And I don't know if they get stressed, but at least they pay attention, and that might be a mechanism.
Well, thanks. That interesting talk, and thanks for the little discussion here about it.
Thank you.
Okay, I'm going to check if we have any other question mark coming in. I don't see any. I'm hoping I don't miss anyone.
No, I think you did fine. Would you like me to stop the YouTube transmission and thank our?
Yes, so I would like to thank Hanja for such a great talk, and everyone for staying here until now. It was a great talk, Hanja. Thank you. We look forward to next seminar next week with Lucy Aplin, talking about a little bit different, talking about cultures in birds, still birds now, moving into different systems. And Hanja, whenever you want to join the community, you're welcome to come over. Thank you, Eduardo, for stopping the YouTube.
Okay, so we're going to finish our